exogenous or endocytic pathway. This pathway leads to presentation of exogenous antigen, which are those antigens that are not made inside the cell that present them on their surface. Antigen presenting cells or APCs. APCs express MHC class II molecules and can present an exogenous antigen to Th cells. This fact is significant because almost all nucleated cells express MHC class I molecules and thus are capable of presenting endogenous antigen to Tc cells, but only APCs are capable of presenting an exogenous antigen to Th cells. Thus, APCs can present antigen in combination with both MHC class I and MHC class II molecules. There are two types of APCs, professional and non-professional antigen presenting cells. Professional APC, non-professional APC. Professional APC in the sense TCs, macrophages, B cells. Non-professional APC, fibroblast, glial cells, epithelial cells, thymus and thyroid, beta cells, pancreas, some endothelial cells. Professional APCs. They express MHC class II molecules and co-stimulatory molecules CD4T and B7. Thus, they can potentially provide both the signals which are necessary for activation of memory Th cells. The first signal is generated as a result of the interaction of peptide MHC class II complex and its cognate TCR. The second signal is generated by the interaction of co-stimulatory molecules. These are the most potent APCs because besides activating memory T cells, they are also capable of activating new T cells. DCs reside in almost all tissues and lymphoid organs. DCs present in the peripheral tissue contains pattern recognition receptors, which recognize the pathogen associated molecular patterns on the antigen. The DCs then internalize the antigen and get activated. This leads to maturation of DCs by upregulation of the expression of MHC class II molecule and co-stimulatory molecules on its surface. Once mature, the DC migrates from the tissue to the lymph nodes, where it can interact with Th cell expressing the cognate TCR and activate it. Since DCs progress components of both innate and adaptive immunity, they are the connecting link between these two types of immunity. Then non-professional APC. Some cells can be induced to express class II MHC molecules and or co-stimulatory molecules under special conditions. For example, during prolonged inflammation, certain cells from some tissues can temporarily take up the role of APC. Antigen presentation by MHC class II molecules. The exogenous pathway of antigen presentation begins with the internalization of exocytosis of the antigen by the APCs. Exogenous pathway, for convenience, pathway has been shown in 9 steps. Step 1 shows the assembly of MHC class II molecule wherein alpha and beta chains of the MHC bind to a chaperone called invariant chain, which helps in their proper folding. Step 2 and 3 represent the transport of MHC class II invariant chain complex from ER to Golgi apparatus and then from Golgi apparatus to the M2C compartment. Step 4 shows cathepsin mediated proteolysis of MHC class II invariant chain complex, leaving behind only clip in the peptide binding groove. Step 5 represents internalization of exogenous antigen by phagocytosis or receptor mediated endocytosis, and then the maturation of phagosome endosome to M2C compartment. Step 6 depicts HLA-DM mediated exchange of clip with exogenous antigen on the MHC class II molecule. Step 7 shows the negative regulation of HLA-DM activity by HLA-DO. Step 8 illustrates cathepsin mediated proteolysis of exogenous antigen to yield peptide MHC complex. Step 9 shows the transport of peptide MHC complex to the plasma membrane. Particulate antigens are internalized by phagocytosis into organelles called phagosomes. Soluble antigens are internalized and pinocytosis or receptor-mediated endocytosis into organelles called early endosomes. Once internalized, the phagosome and early endosome undergo maturation. Maturation involves many sequential changes such as acidification of the lumen and acquisition of new proteins by fusing with organelles or lysosomes of vesicles. Finally, the phagosome and early endosome mature into a special type of late endosome called the MHC class II containing compartment M2C. In the M2C, pH-dependent proteases such as cathepsins perform progressive proteolysis of the invariant chain in the MHC class II invariant chain complex, ultimately leaving behind a peptide roughly 20 amino acid long. This peptide, known as class II associated invariant chain peptide, CLIP, protects the peptide binding groove of the MHC class II molecule. Eventually, CLIP gets replaced by the peptide, thus making the peptide MHC complex. Like invariant chain, the proteolysis of antigen into peptide is also brought about by the cathepsins. Earlier studies led immunologists to hypothesize that antigen is first broken down into peptides, which then bind to peptide binding groove of MHC class II molecule. However, recent findings seem to support a different hypothesis. According to this new hypothesis, the antigen binds the MHC class II molecule as a full length protein and then gets broken down into peptides by cathepsins. The protein HLA-DM facilitates the loading of many antigens by removing CLIP from the peptide binding groove. Further, HLA-DM features the loading of the antigen that have a high affinity for the MHC class II molecule. This HLA-DM performs peptide editing for MHC class II. The protein HLA-DO inhibits the HLA-DM mediated exchange of CLIP with an exogenous antigen. Hence, HLA-DM and HLA-DO have a significant effect on the kind of antigen that gets presented, a phenomenon which has immense immunological implications. Once the peptide MHC complex is formed, it is presented on the surface of the APC, where a passing Th lymphocyte may interact with it through its TCR. In the presence of appropriate co-stimulatory signal, the memory Th cell can get activated by the APC. The naive Th cell, on the other hand, are usually activated only by DC. Then cross-presentation of exogenous antigen. Like all naive T cells, activation of naive T cells is also brought about chiefly by DC. This is possible because of the fact that DC, like most APC, can present antigen in combination with both MHC class I and MHC class II molecules. Does this mean that unless a DC itself is infected with a virus or cancer, it would not be able to activate the new TC cells required to clear the virus or cancer? If so, this can be disastrous for the death of the organism. Fortunately, it is not so. DC can present endogenous antigens derived from other cells of the organism by a process known as cross-presentation. Cross-presentation involves the uptake of an exogenous antigen by phagocytosis or pinocytosis, part of exogenous pathway, followed by its presentation via endogenous pathway. With MHC class I molecule, this phenomenon of exogenous antigens being presented with MHC class I molecule known as cross-priming. Two routes have been hypothesized to explain the events involved in cross-presentation by DC. The first route involved uptake and processing of exogenous antigen in the late endosomes. The peptides this formed are loaded on MHC class I molecule, which are brought into the endosome by recycling from the plasma membrane. The peptide MHC complex is then transported to the surface of the DC. The second route also begins with uptake of antigen using endocytic machinery. The antigen then gets translocated out of the endosome into the cytosol. This is followed by the processing of antigen into peptides with the help of proteasome. The peptides are then translocated into RER in a tap-dependent manner. PLC in the RER loads the peptide on MHC class I molecule. Subsequently, the peptide MHC complex is transported to the surface of the DC. The peptide MHC complex on the surface of the DC is presented with appropriate co-stimulatory signals to a naive TC cell. This leads to the activation of the naive TC cell into a CTL. Two possible routes for the cross-presentation of TCs. Each route is shown using different colors. Blue arrows represent the route which involves internalization of the exogenous antigen, its processing and subsequent loading on the MHC class I molecule inside the late endosome. So brown arrow represents the route involving internalization of the antigen followed by its proteasome-mediated degradation and loading on the MHC class I molecule inside the RER. Both routes result in expression of peptide MHC complex on the plasma membrane of the DC. Not all DCs are capable of cross presentation. The process that enables the DCs to perform cross presentation is known as DC licensing. At first, the DC processes and presents an antigen with MHC class II molecule using exogenous pathway, thereby activating the TH cell carrying the cognate TCR. The activated TH cell then provides the necessary signals and cytokines required for licensing of the DC. This licensed DC can then cross-present the exogenous antigen with an MHC class I molecule and activate a new T cell. On the other hand, cross-presentation by DC can also induce tolerance against self-antigens. This occurs if the licensing by the TH cell is incomplete due to lack of proper co-stimulatory signals or appropriate cytokines. As happens frequently in case of self-antigens, in such situations, the new TC develops tolerance against that antigen. Role of CD1 in antigen presentation. CD1 molecules are a variety of MHC molecules that present lipid or lipid-based antigens. Like MHC class I molecules, CD1 molecules also consist of heavy chain and a B2 microglobulin, beta 2M. The expression of CD1 molecule is restricted to APC and double positive DP cortical thymocytes. CD1 molecules show almost no significant variation between different individuals. Humans have 5 isoforms of CD1 which can be divided into 3 groups, group 1, group 2, group 3. The isoforms belonging to the groups 1 and 2, CD1A and CD1D, are capable of presenting lipid or lipid-based antigens. These isoforms differ from each other in cellular expression, intracellular trafficking, and the kind of lipids they present. The group 3 isoforms, CDI1E, does not present lipid antigens and is a lipid chaperone. CD1 molecule, group 1, group 2, group 3, CD1A, CD1B, CD1C, CD1D, CD1E. Most CD1A molecules on the surface of the APCs can directly interact with lipid-based antigens without requiring the antigen's internalization. On the other hand, most other CD1 isoforms can only interact with the lipid antigen upon its internalization into the endosomes. The mode of presentation is different for each CD1 isoform. The most common pathway is as follows. The APCs expressing CD1 molecule internalize bacteria or aggregates lipid via phagocytosis or endocytosis. Some lipid-based antigens do not undergo any processing and are straight away loaded on CD1 molecule. On the other hand, certain non-peptide antigens are processed inside the endocytic compartments. The processing of lipid-based antigens involve deglycosylation and breakdown of lipid aggregates or membrane structures into monomeric forms. The processed antigens are then loaded on CD1 molecule. The antigen CD1 complex is presented on the surface of APC and is subsequently recognized by cells, NKT cells, bearing the cognate receptor. CD1 molecules show very little variation between different individuals. CD1 molecules show very little variation between different individuals. This fact could have immense impact on the field of vaccine development. This is because MHC loci are highly polymorphic and therefore vary greatly between different individuals. Therefore, while developing a subunit vaccine using peptide antigen, the MHC haplotypes of the entire target population needs to be considered to ensure the efficacy of the vaccine. On the other hand, a subunit vaccine using lipid antigen could be effective for all human populations. However, there is a problem with this approach. Lipids per se are poor immunogens. Further research in this area could provide us with cognate vaccines which could utilize the advantage of both the peptide and lipid immunogens. Now, quick summary. The TCR can recognize its specific antigen only after it has been proceeded to small peptides and subsequently presented with self-MHC molecules. The TCRs of CD4 plus and CD8 plus T cells recognize peptide antigens presented on MHC class 1 and 2 molecules respectively. MHC class 1 molecules are expressed by most nucleated cells. MHC class 2 molecules are expressed by few cells such as DCs, macrophages, B cells and thymus epithelial cells. Endogenous pathway leads to presentation of proteins in association with MHC class 1 molecule. Exogenous pathway leads to presentation of proteins in association with MHC class 2 molecule. Proteosomes are proteolytic machinery present on the cell which breaks down endogenous protein into peptides. So proteolysis of exogenous protein occurs inside the MIAC of or late endosomal compartments and is usually brought about by proteases known as cathepsins. Chaperones and other associated proteins present in the RER help in the proper folding and assembly of the MHC molecule. TAP mediates the transport of peptides from cytoplasm to RER. TCs activate naive T cells by cross presentation of an exogenous antigen on MHC class I molecule. Non-peptide antigens are presented on CD1 molecule.